Welcome guys. Today we will discuss about sodium potassium ATPase. Let's begin our lecture. So the first part is introduction. What is sodium potassium ATPase? Introduction about sodium potassium ATPase. Now this sodium potassium ATPase, it is an integral membrane protein. This is an integral membrane protein. It spans the lipid bilayer. Okay, it spans the lipid bilayer. It is an integral membrane protein. It spans the lipid bilayer and it helps in the transport of 3 sodium out and 2 potassium in. Okay, it helps in the transport of 3 sodium out of the cell and 2 potassium inside the cell. And in doing so, they require ATP. In doing so, they require ATP. So I can say that since they are requiring ATP, so these sodium potassium transporter is known to be a sodium potassium ATPase. Okay, so what I said that it is an integral membrane protein, and this integral membrane protein, which is sodium potassium ATPase, it has 2 subunits. See here I have written that it has 2 subunits and both the subunits can span the lipid bilayer. And this transport is coupled to the breakdown of ATP. When one molecule of ATP is hydrolyzed to ADP and PI, 3 sodium ions are transported out of the cell and 2 potassium ions are taken inside the cell. So what I said that this sodium potassium ATPase, it is an integral membrane protein and it has 2 subunits and both the subunits span the lipid bilayer. In transporting 3 sodium out of the cell, so this is a cytoplasm and this is extracellular region. So they transport 3 sodium out of the cell and 2 potassium inside the cell. In doing so, they hydrolyze 1 molecule of ATP in transporting 3 sodium out and 2 potassium in. So as a result, what we can say that every animal cell has high potassium inside the cell and high sodium outside the cell Now this equilibrium, that is this balance, that is potassium high potassium inside and high sodium outside the cell is required for the normal metabolic function of the cell, and this is maintained by sodium potassium ATPase. If this ATPase would not be there, then the balance of sodium and potassium in a body will be disrupted and there will be several diseases related to it. Okay. So I am again repeating what we studied, that sodium potassium ATPase, they are integral membrane proteins having 2 subunits, and these 2 subunits are alpha and beta. Okay, and both the subunits can span the lipid bilayer. The transport of sodium and potassium is against the concentration gradient, and the transport is coupled to the hydrolysis of ATP. Okay, 1 ATP is when hydrolyzed, then 3 sodium ions are transported out and 2 potassium ions are transported inside the cell. So the outside of the cell is rich in sodium, which is the extracellular fluid, is rich in sodium and inside is rich in potassium. Next we come to in detail about the structure of sodium potassium ATPase. So I already said that this is made up of two subunits. Okay, it is made up of two subunits. It is an integral membrane protein made up of two subunits. So the two subunits, one is alpha and another is beta. So alpha subunit and beta subunit. So two subunits, since one is alpha and another is beta, we can say it is a heterodimer. So one is alpha subunit, one is beta subunit. The alpha subunit is very, very large. It is around 110,000. Okay, and beta subunit is around, it is 50,000 Dalton. Okay, so alpha subunit is very, very large. Okay, and this is the most important subunit. So we will come to the role of alpha subunit. First, what is the role of beta subunit? This beta subunit spans the membrane only once. Both alpha and subunit spans the lipid bilayer. Beta subunit spans the membrane only once. And alpha subunit spans the membrane 10 times. 10 times. So if I draw how it will look like. So see here. Alpha subunit is spanning the membrane 10 times. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so what we can see here, that both N and C terminal of alpha subunit is inside the cytoplasm. And what about beta subunit? Beta subunit spans the membrane only once. Okay, this is a beta subunit and this is the red one is the alpha subunit. So are you getting me? What I said, alpha subunit spans the membrane 10 times and beta subunit span the membrane only once. What is the role of beta subunit? The role of beta subunit is that it helps in targeting. It helps in targeting. So when the proteins are synthesized, okay, after they are targeted to the membrane, okay, so these proteins are targeted to the membrane. So these beta subunits helps in the targeting and proper folding of these alpha proteins, of this alpha subunit. So this beta subunit helps in targeting and proper folding of this alpha subunit and helps in anchoring the alpha subunit to the membrane and helps in proper anchorage of alpha subunit. Beta subunit role is finished. Then the major role is played by the alpha subunit. Now, this in the last slide we discussed that the major role of transport is played by alpha subunit. Okay, so alpha subunit has the major role in transport. Also, the ATP hydrolysis is also performed by alpha subunit. Okay, so the major role of this sodium potassium ATPase is performed by alpha subunit. Now, since they require ATP for the transport of ions and since they require ATP for the transport of ions, this process is known to be as active transport. And the ATP is directly required. Since the transporter is directly requiring the ATP, It is known to be as what type of transporter? It is known to be as primary active transport. Now, can you tell me one example of primary active transport? So, it is sodium potassium ATPase, where ATP is directly required. Okay, so this is why they are known to be as primary active transport. Next is this 3 sodium are moving out of the cell and 2 potassium are coming inside the cell. Okay, so as a result, what happens is the cell from inside the cell, there is the net movement of one positive charge out of the cell. Okay, 3 positive are going out and 2 positive are coming in. So there is a net movement of one positive charge out of the cell. Okay, so we can say that the outside of the cell is positive. So again, if I draw, 3 sodium is going out and 2 potassium is coming in inside the cell. So there is a net movement of one positive charge outside the cell. So comparatively, we can say that the inside of the membrane is negative relative to the outside of the membrane. Okay, we can say that the inside of the membrane is negative relative to the outside of the membrane, relative to the outside of the membrane. So we can say that when these pumps are working, they create the outside positive and inside negative. As a result of which, due to the working of these pumps create potential difference. These pumps create potential difference. That is why these pumps are known to be as electrogenic pumps, because they are creating the potential difference. That is why they are known as electrogenic pumps. So I repeat what I said. I said that if we see that these ATPase transport the 3 sodium out of the cell and 2 potassium inside of the cell. Okay, so they are creating the potential difference by moving out more positive out. Okay, more positive ions are moving out of the cell, so they are creating a potential difference. Since they are creating the potential difference, they are known as electrogenic pumps. Again, important thing, another important thing which I said, that this alpha subunits, they are mainly involved in the transport of sodium and potassium. So these alpha subunit of the protein, they have the potassium binding site, they have the sodium binding site, they have the ATP binding site. So all the proteins, all the major proteins, ions which are required to bind and to be transported, they have their binding site in the alpha subunit only. Beta subunit role is very simple, that is just targeting folding and proper anchorage of alpha subunit to the membrane. Okay, and alpha subunit will have the binding site for sodium also, potassium also, for ATP also. Next, now this sodium potassium ATPase, they exist in 2 conformation. One is the phosphorylated conformation and another is dephosphorylated conformation. Okay, so if I summarize, see here, So steps, if I write, so I will explain the last slide after this slide. So here what it is, see, the first step is at first, in the first step, that ATP will bind to the sodium potassium transporter. At first there is a binding of ATP to sodium potassium ATPase. Okay, this sodium potassium ATPase, when they bind with ATP, then they have the higher affinity for sodium. What I said, that when sodium potassium ATPase, when they bind with ATP, they have higher affinity for sodium. So after which sodium will bind. Now, when sodium will bind to the sodium potassium ATPase, okay, then ATPase activity of the sodium potassium ATPase will come into play. ATPase activity will be seen where the terminal phosphate, in which the terminal phosphate of ATP is transferred to aspartate residue of this transport protein, where the terminal phosphate of ATP is transferred to aspartate residue of transporter protein. Okay, here the transporter protein refers to sodium potassium ATPase. And after this ATP hydrolysis, there will be the release of the sodium. The conformational change is seen in the transporter protein. I will show you the picture, after which the sodium is released outside. Okay, so sodium is when released outside, then there is the binding of potassium. Okay, once potassium binds, then there is dephosphorylation. There is dephosphorylation of this aspartate residue, and dephosphorylation also causes the conformational change. So dephosphorylation followed by the conformational change again. So when there is a conformational change, then the K plus will be released inside. So this I will explain you with one picture. So see, so this is a picture of this model of sodium potassium ATPase. In first case, what we see, so here what we see, that when sodium binds, then ATP will be hydrolyzed. Okay, when sodium binds, then ATP will be hydrolyzed. Hydrolysis of ATP causes the conformational change. Okay, and due to this conformational change, the sodium are released outside. Okay, here for simplicity, there is the binding of only one sodium and one potassium is seen, but actually it will be there will be the 3 binding sites for sodium and 2 binding sites for potassium will be there. Okay, for simplicity here, only for one ion is being shown. So see here, so after the conformational change, the sodium will be released outside. Then there will be the binding of potassium. Okay, number 4, what we see, that there will be the binding of potassium from the extracellular surface, and after the potassium binds, there is dephosphorylation. Okay, after the dephosphorylation, what we see, that the protein return to its original conformation. So again I repeat, in the first stage there is the sodium binding, then there is ATP hydrolysis. This ATP hydrolysis causes the conformational change and causes the sodium to release outside. 3 sodium will be released outside. After the sodium releases, potassium will bind in. Okay, there will be the binding of potassium from the extracellular surface, and there will be the dephosphorylation after the potassium binds. Okay, then the protein will return to its original conformation when it transfers the potassium from the extracellular side to the cytosol. Is that clear? Now one important thing I'll again discuss, that whenever I'll change the color, then it will be more easier for you to understand. I'm taking a purple color. So see here, in the first condition, this conformation, in number 1, the conformation is shown. This conformation is known as E1. Okay, and in the E1 state, the transporter is bound with ATP. Okay, so this is the E1 conformation where the transporter is bound with ATP, and this E1 conformation have high affinity with sodium. It has high affinity with sodium. Okay, now after the ATP is hydrolyzed, this E1 state changes to E2 state. After the aspartate residue is phosphorylated, okay, after the phosphate residue is phosphorylated, this form is known as, I'm sorry, it should not be here. This conformation changes to E2 conformation. Okay, and this E2 conformation, it has high affinity for potassium. Am I able to explain you what I said? That this sodium potassium ATPase, they exist in 2 conformation. One is phosphorylated one, another is dephosphorylated one. Okay, the dephosphorylated one is the E1 state. The E1 is dephosphorylated, and the dephosphorylated one have high affinity for sodium. And once ATP is hydrolyzed, once they are phosphorylated on their aspartate residue, this E2 conformation is formed. The E1 is flipped to E2. Now this E2 have high affinity for potassium. It will bind to potassium. The phosphorylated state will have high affinity for potassium, and this phosphorylated state is known as E2. Now this dephosphorylation state is E2, which has high affinity for potassium and low affinity for sodium. So see here. So what I said, that there are 2 conformations. One is phosphorylated conformation, another is dephosphorylated conformation. In the phosphorylated conformation, okay, it is E2, and in the dephosphorylated condition, it is E1. Okay, this E1 have high affinity for sodium ions, and this E2 have high affinity for potassium ions. Okay, so first of all, if I again write the steps, so what will happen, that E1 and ATP are there in the first case, and they have high affinity for sodium. Okay, they have high affinity for sodium. E1 plus ATP, they have high affinity for sodium, and when this ATP is hydrolyzed, what will be formed? When this ATP is hydrolyzed, then it will be enzyme 2 plus ADP will be released. Again, this phosphorylated state will turn back. Here I write E2 simply, that will be more easier. Then this E2 will again be converted to E1 state upon dephosphorylation. Okay, upon dephosphorylation, it will be converted to even again. Now this will be continued in the next class, and till there I have attached one animation with the video. Please watch it. Thank you.
The sodium-potassium pump is an active transport mechanism that is driven by the breakdown of ATP and works through a series of conformational changes in a transmembrane protein. 3 sodium ions bind to the cytoplasmic side of the protein, causing the protein to change its conformation. In its new conformation, the molecule becomes phosphorylated at the expense of a molecule of ATP. The phosphorylation induces a second conformational change that translocates the 3 sodium ions across the membrane. In this new conformation, the protein has a low affinity for sodium ions, and the 3 bound sodium ions dissociate from the protein and diffuse into the extracellular fluid. The new conformation has a high affinity for potassium ions, 2 of which bind to the extracellular side of the protein. The bound phosphate now dissociates, and the protein reverts to its original conformation, exposing the 2 potassium ions to the cytoplasm on the inside of the cell. This conformation has a low affinity for potassium ions, so the 2 bound potassium ions dissociate from the protein and diffuse into the interior of the cell.